Hi everybody, welcome back to the Experimental Design and Analysis section of the Core Skills module. So remember, this is a year-long Core Skills module, and we, before Christmas, got about halfway through the content of this Experimental Design and Analysis section. So after Christmas now, we're going to look at the last few topics and start to think about preparing yourself for the exam. So there's an exam in May, June, which will be a ROGO exam assessing everything that I've taught you in the lectures. That's separate from the sort of more practical side of the experimental design and analysis section of the course of Core Skills. That's very variable in depending on which degree you're doing. So the biology students are doing something with me, a workbook. The neuroscience students are doing something with Paul Smith, using a different software package, etc., etc. So you need to talk to your individual module convener or look at the Moodle page for your degree to understand how the practical aspects of sort of learning to analyse data will happen in Core Skills, and that's well underway for most of you. Whereas for the theory, you come to me in the lectures, so that's why you're here now. Okay, so today's session is about counts and correlations. You know, I'm always a bit desperate to find decent photographs of decent images to illustrate the concepts I'm talking about, so I've actually had to resort to Sesame Street here. Apologies for that. But we're going to talk today about really two quite distinct topics, but each of them is relatively small, so I wanted to deal with them together in one lecture. So first we'll look at counts and then we'll look at correlations. So the outline for the lecture as a whole is firstly to address this learning outcome, which is to be able to perform and interpret a chi-squared test. Some of you will have heard of a chi-squared test, some of you will have done one before, but for those of you who don't know about it, it's a test which allows us to look for differences in frequencies. So we're going to look at frequencies which emerge when we count some kinds of data. I'll explain more in a moment. So that's the first job. And then we're going to look at trends. So this is moving on. In many of the last lectures, we've looked at differences, biological differences of some sort, and we looked at how to test two samples to see whether they have the same or different mean using a t-test, for example. Today we're going to look at frequencies, but then in the second half of the lecture we're moving on to a really quite different topic, which is looking for trends. So this is where we're looking for correlations or associations between pairs of continuous variables, and we're going to do that using two approaches, something called correlation and something called regression. And in today's lecture I'm going to look at the difference between those two approaches and then focus in on correlation to start with, and we're going to look at how to do a kind of parametric correlation test and then how to do a non-parametric correlation test. And then later on in the course, not in the next lecture but a few lectures time, we'll come back and look at regression. Okay, so part one then, it's quite a long part this for the lecture, and there'll be a shorter part later on. It's just the way that the content splits up. We're going to look at the chi-squared test and how it can be used to test for hypotheses about frequencies. Before I go on though, just I mentioned the exam already, and I'm sure those of you who've got through the January exam period, having had exams for some modules, will be relieved to have them behind you. You may have already noticed that there is a mock exam available for this module for the experimental design and analysis content. Apologies it wasn't available a bit earlier, but I managed to make it available I think about a week ago. So if you haven't seen it yet, go onto the Moodle page for the generic Core Skills page, and you will find a link to the mock exam. Actually, I've just realised I've put the link in the biology Core Skills page, so it's all right, I will update that this afternoon so that it's available to everybody. So the mock exam that I've created, you should be able to link through to it at any time over the next few months, so there's no particular time pressure to do it. Initially it covers lectures 1 to 6, so all the content that I delivered before Christmas. It's using the same kinds of question format that you're going to get in the real ROGO exam, with a similar kind of timing, so in terms of how much time pressure there'll be to get through the questions in the time. So do go and have a look at that. You can come back to it more than once, I think. When you've done it, you can get some nice feedback about the different learning outcomes that the questions relate to, so you can get a kind of traffic light feedback about whether you've understood particular learning outcomes. Although with a relatively small sample size of questions, there aren't that many questions that relate to each learning outcome, so in a sense it's kind of obvious. But what I'm hoping to do is to put in place a further mock exam later in the semester which covers the remainder of the content for the course, so that by the time you get to the real exam in May, June, you will have had a chance to practice your understanding of the whole set of lectures, as well as the sort of understanding what kind of question format I use. Now, every different degree has a slightly different form of assessment for this core skills module, and so I noticed that Paul Smith, the neuroscientist, had created a neuroscience specific version of the mock exam. I'm not sure he's made it live for this year yet, but he will be doing, I'm sure, in due course. And that is very similar to the main mock exam that I've just explained, but it adds a few questions which relate to using Prism, that's the software that you've been using in the neuroscience degrees. So just to explain then that as well as the sort of general mock exam, it may be, at least for neuroscience, maybe for some other degrees, that your lecturers decide to put in some extra questions if you're likely to get assessed on that content in the final exam in May, June. But the critical thing, as ever with core skills, I know it's a complicated module, is to go to your, like the Moodle page for your own degree to check out exactly what you need to do if you're a sports and exercise science student or a neuroscience student. The generic module contains stuff that everybody does, but that's not everything that you will be doing. So you do need to follow the instructions on your own degree page, and it will sometimes refer you to the generic page where you'll get my content and my mock exam. I hope that's all clear. So let me know if there are any problems with that mock exam. It's always slightly difficult for me to set up because I can't quite see what you can see in Moodle and Rogo, but I think I've got it working, fingers crossed. So good luck with that. Okay, so we're going to spend the first part of this lecture looking at differences, but rather different kinds of differences from those that we've encountered in earlier lectures. So if you remember, we spent quite a lot of time last semester looking at this kind of scenario where we have two continuous samples or samples of continuous data that we want to compare because we think there might be a difference in the mean of those two samples. So here we have a green and a blue sample, which might be, I don't know, the heights of men and women, and we want to know, are men taller than women? And we developed a testing framework, a statistical testing framework, in order to address that question. But there's a whole set of other kinds of difference scenario that we encounter in the life sciences where we can't use those sorts of tests that we've just developed because the underlying data are not truly continuous. And if the data come in the form of frequencies of categorical events, so the underlying data are categorical, they fall into categories rather than on a continuous numerical scale, then when you want to look for differences, you end up with a really rather different situation. You can't actually really draw a frequency histogram like this. You can't imagine a bell curve if what you're doing is, if the underlying raw data are categorical. If I'm looking at individual humans and I'm scoring them as either male or female, I end up with a frequency of males and a frequency of females. And I might want to compare those frequencies to find out, for example, if the sex ratio is equal or if there are more men than women. And as soon as I get into that scenario, I've now got essentially a collection of frequencies indicated by F here, which I'm looking for a pattern in, but they don't, it's difficult to imagine them on a frequency histogram as a continuous measure. So we're going to need to develop a different framework for assessing questions about these kinds of differences, and that's what we're doing today. So the continuous variables, just to remind you about what we were doing before Christmas, I know it'll feel like a long time ago. We have been looking at a whole bunch of different examples to illustrate the sorts of situations where we want to look for differences in continuous variables. So we looked at a whole load of examples based around sperm swimming speed, for example, where we were measuring speed on a continuous variable. Similarly, we looked at several examples where time was being measured, or the colour of urine even, which you can measure on a continuous scale. How blue is your urine? Etc., etc. So these were all kind of classic examples of continuous data, and then we were looking at two or more samples, asking, are those samples different? But very often the data we have are not continuous, they're actually discrete. The data fall into discrete bins of some sort. Sometimes they fall into nominal categories with different names, but sometimes actually they fall into sort of numeric categories that we can actually treat effectively as if they're continuous. So there's a bit of a distinction here. There are two kinds of scenarios where we have discrete data, and in one scenario actually we can basically use the approaches that I've talked about before. So the most common form of discrete data that you're going to encounter in the life sciences come in some form of count or frequency, and count data in a kind of classical sense can be most of the time treated as if they are continuous, specifically if we collect whole samples of count data. So what do I mean? Well, so here's three things that we could count. We could count cells down a microscope, we could count bed bugs in your bed or ants in an ant's nest, and we end up with a number, 300 ants or 200 cells or whatever it might be, which is numeric and can be treated as if it's continuous, even though you can't have half an ant or half a cell if that's what you're counting. And in fact, you can't have negative cells or negative ants. So although the fundamental here, the underlying data are discrete, they come in integers, you can't have fractions of ants or cells, you can, under most circumstances, treat them as being approximately like continuous data. And in particular, when you're collecting samples of count data. So by that I mean if you've got lots of count measurements in each group that you're interested in. So if I'm interested in whether there are more ants in the ant nests of one species compared to another, and I've gone to lots of ant nests and counted the ants, then I'm going to end up with a whole sample of counts with which I want to compare another sample of counts, two different species, two different samples of counts of ants. And the same goes for cell counts down a microscope or in a cell fight. My brain has gone blank. That thing that you use for counting cells, flow cytometer. That's what I'm thinking of. So if you're using a machine to count cells, maybe you're looking at whether patients with a particular disease have more of a particular type of cell than patients who don't have the disease. If you've got lots of samples of counts, lots of individuals from each of which you've counted the number of cells, then we can consider that, we can approximate that by thinking about two continuous samples of data that we can compare using a t-test or something similar. So that all sounds fine. There's a difference though. There are situations in which we have frequencies which can't be collected together in samples and compared in the way that we've previously done. So think about these two examples. With human beings, you might be interested in maybe human genetic traits which might affect their health, or just might be a bit weird, like the fact that your ability to roll your tongue, I think, is influenced by your genotype. And so some people can roll their tongue and some can't. You can, for each individual person, you can ask whether they can roll their tongue or not. But if your samples come in the form of collections of individuals, each of which falls into a category, so each person is either a tongue roller or not, and if you want to know, for example, whether more people can roll their tongue than can't, then you've got a collection of individuals that you can put together to create a count of tongue rollers, and a collection of individuals that you can put together to create a count of non-tongue rollers. You end up with frequencies that you're interested in comparing, the relative frequency of being able to roll your tongue and not. Same goes for sex, for example. So in peacocks, males and females, we might be interested in whether there's an equal sex ratio, or maybe whether one species has a different sex ratio from another species. If each individual yields a single nominal data point, it's either male or female, we can top those up in the forms of frequencies and compare them, but we need a very different kind of test. So we're going to work through an example of this. I mean, trying to describe it in abstract terms, as I've just done, is actually quite hard, and you probably maybe got a bit confused about the way I was explaining it. It's much easier when you see a real example to understand what we're trying to do here. So we're going to use an ecological example. I'm an ecologist, I'm afraid. Sorry, you have to put up with an ecological example, but it turns out to be quite interesting later on. Initially, this initial example is very simple, but I'm going to develop it later on. So we're going to look at blue tits, familiar garden bird. I'm sure you've all seen them. You maybe haven't stopped and thought about just how spectacularly beautiful a blue tit is, and in particular they have these amazing bright blue plumage, which is relatively rare in the natural world, and that's going to turn out to be significant in the story that I'm going to tell you. But initially, let's just imagine we're interested in sex ratio in blue tits, because it gives us a really simple example of why we might want to compare frequencies. So we might just simply want to know whether we have an even sex ratio, 50% males and females, or whether there's some kind of sex bias. And it turns out in the natural world that some populations do show a male or a female sex ratio bias for lots of different reasons. So, for example, there are these weird bacteria which can kill males, so they make populations female biased, which creates all kinds of interesting issues. You can imagine if there's a lot more of one sex than another, then suddenly finding a sexual partner becomes really difficult. So, I mean, that's one of the main reasons why the default is we expect a sex ratio to be equal. But let's imagine we've gone out there in the natural world, looked at a species, in this case blue tits, counted the numbers of males and females, and we want to know whether the null hypothesis that there's no difference between the numbers or the frequencies of males and females is true, or whether that null hypothesis should be rejected in favour of an alternative hypothesis that says there really is some kind of bias in the sex ratio. So I'm going to sample a bunch of blue tits, and I find I've got 103 males and 88 females. And you look at that and you think, that's more males than females, but is it more than we'd expect at random by chance if the null hypothesis is true? If you remember from before Christmas, that's the fundamental idea when we do statistical testing. What we look at is data, and we ask how likely are the data to have arisen if the null hypothesis is true. And the answer to that question is always the same thing, it's p, the p-value. If the probability of these data under the null hypothesis is very, very low, so low that we can kind of ignore it, then we might conclude the null hypothesis is wrong, and in fact there is a difference in the sex ratio. In this case, it's really hard, I think, to look at those numbers and reach a firm conclusion intuitively. That's why we need stats, because we need an objective rather than a subjective opinion about whether this difference we can see, slightly more males, is kind of real or not. So to test the null hypothesis, we're going to use this thing called a chi-squared test. So the Greek letter chi looks a bit like an X, but one limb is straight and the other is curly. And so chi-squared is the test statistic in a chi-squared test, and we're going to look at how to calculate chi-squared and then to generate a p-value from it. So the way that this test works is to compare the observed frequencies with what we'd expect if the null hypothesis is true. And then that allows us to calculate the statistic, chi-squared, and from that a p-value. So for a given particular sample size and a particular value of chi-squared, then we can generate a particular p-value. In other words, for every given sample size, there is a particular probability of observing a value of chi-squared of a certain number if the null hypothesis is true. So let's look at this example. Here are some observed data. We've already seen those, 103 males and 88 females. And now we have to work out what we would expect if the null hypothesis is true. So, well, what would we expect if we have equal numbers of males and females and we go and sample this many birds, 191 birds, then obviously we'd expect 95.5 to be male and 95.5 to be female. So immediately you spot that that doesn't make a lot of sense because you can't see half a blue tit. But actually, in the world of stats, that's perfectly fine. On average, if we continued to do this experiment over and over again, we would expect to see 95.5 males and 95.5 females. So that's our expectation under the null hypothesis, and it's that we're going to compare with the reality, what we actually observed. And we're going to calculate this chi-squared statistic using a simple equation which will look slightly familiar because it's kind of similar to one that we've already encountered in this course. So chi-squared is the sum of the observed data minus the expected squared divided by the expectation. And we're going to add these numbers up for every single frequency that we have in the data set. So from the first count or frequency up to the last count or frequency, and here the number of frequencies that we have in our data set is labelled A. So you might be slightly confused because you're maybe expecting n to be the sample size. Well, what we need here is to distinguish between the total number of birds that we've looked at, which we would typically call the sample size n, in this case 191, or 88 in one sample and 103 in another. And A, the number of counts. In this case, we just have 2 frequencies or 2 counts in our data set, a male observation and a female observation. So we're going to do a very simple sum here across 2 numbers. We've got 2 observed data points, and for each of them we're going to calculate what this equation is. So we take each observation, so 103 males is the first observation, for example. We're going to take away the expectation, square it, and divide it through by the expectation. So yeah, I've already said all of that. Okay, so let's actually look at that. It looks a bit horrendous at first, but it's actually not too bad. So we've got 103 males, remember. So for the first observation, we take away the expectation, and the expectation was equal numbers of males and females, 95.5 males and females. So we look for the difference between what we've seen and what we'd expected if the null hypothesis was true. We square it and divide it through by the expectation, and then we add that, using the sum sign, to the next difference between the observed number of females this time and the expected number of females, squared, divided by the expectation. Now this looks very familiar, if you recall, to the sum of squares, where we looked at the difference between the mean and an individual data point, squared it and divided through by the sample size. So the principle here, what we're doing is quite similar, because we're taking a difference, because the difference between the observed and the expected, that's sort of quantifying how weird our data are if the null hypothesis is true. So if we see a huge difference, then that means the data look pretty weird if the null hypothesis really is true. Whereas if the 2 numbers are very close together, that suggests that the data are quite consistent with the null hypothesis. So that's why we're taking this difference, and we're squaring it to get rid of the negative sign, because sometimes, well, as you can see, if there are more males than females, we get a positive difference. If there are if there are more observed birds than expected, we get a positive difference, and vice versa. Here we get a negative difference, but it's still a difference, and we're interested in how big is that difference. So we square it to get rid of the minus sign, and we scale it by the expectation by dividing through by the expectation. You don't need to worry too much about the workings of this equation. I'm showing you it here, but in real life as a scientist, what you're going to do is you're going to plug this into a computer, and it's going to tell you chi-squared, and then from that you can calculate a p-value. But it's important to understand the principle at work here, which is basically we're trying to quantify how like the expectation the observed data really are. And you get a number at the end of this process, which doesn't mean a lot. It's not in obvious units, but it follows what we call a chi-squared distribution. So 1.18 is the number here, and for a given number of counts or frequencies that you've got in your data set, that chi-squared has known mathematical behavior, and so we can from it calculate a probability or a p-value, the likelihood that we would see that kind of number if the null hypothesis is really true. And this number, just like most stats tests, is going to get bigger the weirder the data are, or at least the less consistent the data are with the null hypothesis. So as with most stats tests, there's another thing you need to know before you can calculate your p-value, which is the degrees of freedom, and that reflects something about the power of the test, the size of the study, if you like. And in the case of a chi-squared test, it is the number of counts or frequencies minus 1. And in this very simple example, we only had 2 observed frequencies, a equals 2, so the degrees of freedom is 1. Once we know the degrees of freedom and the chi-squared value, we can then plug that into a computer, or in the old days you'd actually look it up in the back of your stats book and find out what the p-value is that corresponds to that chi-squared value. So I've done that for you here, and certainly the biology students will have a go at doing this. You can do it in Excel, and I think the neuroscientists, for example, might well do it in Prism. So in this case, we get a chi-squared value of 1.18, degrees of freedom 1, and a p-value of 0.28. So that means there's a 28% chance of seeing a chi-squared value of at least 1.18 if the null hypothesis is really true. So that's quite a high chance, certainly not low enough that we want to reject the null hypothesis. So we conclude that the sex ratio that we've seen, although there were slightly more males than females, is not significantly different from a 1-to-1 sex ratio, or equal numbers of males and females. So at the moment, we don't have any evidence to think that this blue tip population is in any way unusual. It's conforming to expectations, really. We would expect animal populations in the wild to have equal sex ratios. So happy days. So chi-squared test is quite flexible and it can deal with lots of different situations, and we're going to look at a rather more complex situation in a moment involving those blue tits. But just to show you an example of a subtle variation on this theme, because chi-squared tests can cope with other kinds of hypotheses. In the case of the sex ratio, we were simply asking were there equal numbers of males and females. But there are other situations where the expectation under the null hypothesis is not 50-50, but is some other combination of frequencies. And a really typical place where we see this in the life sciences is in genetics. So if we look at a kind of classic Mendelian cross of some pea flowers, you've all learnt about this, I'm sure, at school, in A-level biology, you'll know that if you cross two heterozygotes where this locus R is controlling the colour of these pea flowers, then you can get a bunch of different offspring with different genotypes. And if this locus is important for controlling the colour of the flower, we can see different coloured offspring. So imagine that we cross two red pea flowers, and capital R is the dominant allele here. So if you're heterozygous, capital R, little r, you've got one copy of the dominant allele. I'm going to say in that situation you have red flowers, because the lowercase r stands for the recessive allele, which in this case is going to give you white flowers, but only if you're a homozygote. So we're going to cross two heterozygotes, look at the different possible outcomes, and come up with an expectation which generates our null hypothesis. So the four possible offspring genotypes, or F1 genotypes as they're known in genetics, are like this. So, because if you take one chromosome from each parent and mix them up at random, you're going to come up with four possibilities. Either you're going to have flowers which are homozygous for the dominant red allele, or homozygous for the recessive white allele, in which case you'll actually get white flowers, or you'll have more heterozygotes in your offspring, and each of those will have at least one, or have exactly one dominant red allele, so you're going to end up with red flowers. So in this situation, if we're correct about the genetics, then when we cross two red flowers, we should see a ratio of 3 red flowered offspring to 1 white flowered offspring, because only a quarter of the offspring, those that happen to be homozygous recessive, are going to have white flowers, and all the rest have got at least one dominant allele, so they're going to have red flowers. So here what we're going to do in this little very simple experiment is test whether we're right, whether this Mendelian system is really working in these flowers, or if something strange is going on. Maybe we've got the genetics wrong, maybe there's more than one locus involved, some other alleles getting in there and complicating the situation. But we can test that using some data by counting the offspring. So we're going to test the null hypothesis that the offspring, or F1 ratio of red to white flowers, will not be different from 3 to 1, 3 red to 1 white. So we do our cross. We patiently wait for the seeds to grow, and then they produce flowers, and then we can see that we've got 38 red flowering peas and 22 white flowering. So you think, okay, well, that's pretty consistent with what we're expecting. We've got more red than white, so that suggests that our hypothesis about the genetics is probably correct, isn't it? Well, obviously it would be dangerous to reach that conclusion just by eyeballing the data. What we need is a formal assessment of this null hypothesis using a p-value. So to do that, we're going to do a chi-squared. I won't take you through too much of the gory detail, but the principle is exactly the same as with the blue tits. We've got observed numbers of red and white flowers. We've got expected numbers of red and white flowers, which are generated by the null hypothesis, which says we expect a 3 to 1 ratio. So we're expecting 75% of the flowers to be red and 25% to be white. Given that in total we've got 38 plus 22 equals 60 flowers, 75% of 60 flowers is 45. So we'd expect 45 of these plants to have red flowers and 15 to have white, which is a little bit different from what we've seen. So the observation is a bit different from the expectation. Is it sufficiently different that we have to reject our null hypothesis? And the answer is, well, we can generate the value for chi-squared, the degrees of freedom, which is the same as before, and from that then we can get the computer to tell us what the probability is. And the probability here is actually only 4%. So what we're seeing is we should have 3/4 red. We've actually got quite a lot less than 3/4, and it's sufficiently less, given the sample size, that that's actually really unlikely to happen, only a 4% chance if the null hypothesis is true. So in this case, we actually reject the null hypothesis, even though it wasn't necessarily blindingly obvious from the data. The data are actually quite unlikely under the null hypothesis. So we're going to conclude that our hypothesis about the genetics of these plants is somehow wrong. We need to go back to the drawing board and figure out what is going on. Maybe there's another allele here that we didn't know about. Maybe we were wrong about the dominant recessive relationship or something else. Okay, so that shows you that a chi-squared test can deal with frequencies when you have an expectation, even if the expectation is not equality. But chi-squared test can deal with more complicated situations, and so I want to look now at what we call a two-way chi-squared test, where we have two variables of interest working at the same time, and the hypothesis relates to the interaction between those two variables. So we're going to go back to our blue tits, and the reason I introduced the sex ratio in blue tits is because I wanted to tell you a story about this really cool paper that appeared in Nature quite a while ago now, about a decade ago, looking at the role of this bright blue plumage in blue tits. And it was thought, hypothesized, that the blue plumage was sexually attractive, and it indicated to females that you're a high quality, fit and healthy male that's going to produce really good offspring. So that was the kind of underlying theory behind this experiment. And what they did was they looked at what happened when females mated with bright blue, shiny, lovely blue tits, males, and then those that blue tits that didn't look so good. And what they wanted to know was if a female mates with a dodgy looking blue tit, a dodgy looking male, does she change the sex ratio of her offspring? Because we know from some animals in the wild that some females are able to control the sex ratio of their offspring. In other words, to manipulate the relative frequencies of males and females in their, well, in the case of blue tits, in the clutch of eggs that they lay. Why on earth would you do that? Well, the hypothesis goes that if you're a female and you mate with a nice, bright, beautiful male who is fit and strong and sexy, likely to attract other females, then it pays you, potentially, to have lots of male offspring with that male. Because if you have male offspring with where the father carries these really good genes that give rise to lovely bright plumage, then your male offspring are going to be really sexy and successful, probably going to attract lots of mates and have lots of babies themselves. So it's in their evolutionary interest, the females, to bet in favour of male offspring if you know that the father is a real good one. By contrast, if you've got stuck with a rather dodgy looking male and he's going to be the father of your eggs, your offspring, then maybe, the hypothesis goes, you'd be better off having female offspring, because females don't need to use this blue plumage to attract mates, and so, because apparently that's how it works in blue tits, so you kind of it's the best of a bad job. Given that you've got a dodgy father whose genes code for rather rubbishy plumage, you're better off having female offspring where it doesn't matter. So this was it sounds like quite a far-fetched hypothesis, but actually the more we know about sort of the way that females can control their offspring phenotypes, it's not entirely implausible. And so an experiment was done to test this idea. So the fundamental of the experiment is quite simple, really. We're looking for whether the sex ratio produced by females, sex ratio of her offspring, is affected by what the males look like, the males that they mated with. So we've got two things that are varying in the experiment. We've got what the males look like, what we'll call male quality, or how bright blue their plumage is, and then we've got the sex of the offspring, are the offspring male or female. So the underlying question is, do females lay more eggs, more male eggs, when they have mated with an attractive male? And the experiment is quite clever, because it takes a bunch of male blue tits and it forces half of them to look dull and like not very sexually attractive. And the way that you do this with a blue tit, it turns out, is as simple as smearing it with sunscreen. Because it turns out that the deep blue signal that's really important in attracting female blue tits is in the ultraviolet end of the colour spectrum, and that if you put sunscreen on to the feathers, you can't really see that sunscreen to the naked human eye, but to a female blue tit, what you're doing is blocking out the bright UV reflectance from the plumage. So the female, looking at a male covered in sunscreen, sees a dull blue male that's not attractive. Whereas in the control birds in the experiment, where they didn't apply the sunscreen, the plumage appears relatively bright and blue. So the experiment involved producing two groups of male blue tits that were different in their sexual attractiveness because we'd experimentally manipulated them. And the null hypothesis was that the sex ratio in the clutches of eggs from the females that had mated with the control males that hadn't been manipulated would be the same as the sex ratio in the clutches of eggs produced by females that mated with blue tits that had been covered in sunscreen. And if we see a departure from that null hypothesis, that would be consistent with our alternative hypothesis, which is that actually females are able to manipulate the sex ratio of their offspring in response to the quality of their mate. So to keep things simple, this is a real experiment, but I've taken some sort of example data or simulated data, and to keep things really simple, we're just going to look at the first chick in each clutch produced by the females, and this is going to avoid us having a problem of pseudo-replication. If we looked at all of the eggs in a single clutch, when they share the same mother, then those data points won't be independent because they all share the same mother. But you don't need to worry about that too much. We're just going to get a bunch of nests, take the first egg out and ask, is the chick that hatches male or female? Oops. So we end up with what's called a 2x2 contingency table. So this is when you have lots of frequencies like this counted across more than one variable. We call it a contingency table. And so here we've got the one variable of interest, which is what sex are the chicks, are they male or a female? And then the other variable of interest is whether the male, the father, is good, in other words, not covered in sunscreen, or poor, covered in sunscreen. So we do this. Let's hope we've got 200 nests with blue tits, and we tot up the first egg in each nest, and we find that overall we've got 106 females and 94 males, so slightly female biased overall, but not massively different. But if you look at the female birds that were mating with the control unmanipulated good quality males, you find actually there's slightly more male chicks being hatched, 56 as opposed to 44. Whereas where the males were covered in sunscreen, you're seeing, getting on for twice as many female chicks as males. So it looks a bit like the hypothesis we have in mind that mate quality affects sex ratio could be working here. But again, eyeballing the numbers, it's hard to tell what the probability would be, what the p-value would be of the data under the null hypothesis. So let's do a chi-squared test. So it's just like before, we've got observed data and then we've got an expectation under the null hypothesis. The difference is that the expectation is a little bit more complicated. So the observed data we've already seen, they're straightforward enough. To calculate the expectation, what we need to do is say, well, what would happen under the null hypothesis? The null hypothesis says it doesn't matter whether the males, the fathers, are good or poor, you'll end up with the same sex ratio. But that's not the same as saying that you'd expect the sex ratio to be equal, 50% males and 50% females. And this is really important, this is the thing to spot, that in this particular experiment, we've actually got slightly more females than males overall. So we have to take that into account when we generate the expected frequencies of males and females in the nests of birds that have mated with good and poor fathers. So what we do is we get the relative frequency of male chicks overall, which is 94 out of 200, so 94 of all of the 200 nests produced male chicks. So we take that frequency and we multiply it by the total number of birds that mated with control good quality fathers, in which case, in this case it was 100, because we had 100 actually birds that were given good quality mates and 100 that were given poor quality mates. So we say of the 100 that were given good quality mates, we'd expect 94 divided by 200 as a ratio of the chicks to be male. So we do that, we add that up, and that allows us to generate an expected frequency of male chicks in nests from good quality fathers. And we can do the same for females, and then we can do the same for the poor quality treatment as well. So what do the answers look like? Well, what we're going to get is an expectation that in the good quality father treatment, we should have slightly more females than males. Why is that? Well, that's because there are slightly more females than males in the whole population. We don't care about why that is, because we're interested in whether this expectation that the quality of your father doesn't affect your sex ratio is true or not. And so if the quality of your father doesn't affect your sex ratio, then we're going to expect a slight female bias because that's what the population's like. So we can do the same calculation for the poor quality fathers, and you should see the same ratio, got the same number of birds in total, 100, so we're going to expect exactly the same pattern for both the poor and the good quality males, because that's what our null hypothesis says. It says that there's no difference. It doesn't matter what your father looks like, you're going to have the same sex ratio offspring. And then this gives us our complete set of expected data to compare with our observed data. So we've got 4 counts of observed data and 4 counts of expected data. We can plug those into our equation. So you take the first observed data point, take the difference from the expectation, divide through by, square it, and then divide through by the expectation. Add that up, do that for all 4 counts, and hey presto, you get a chi-squared value, in this case 6.5. Then you need the degrees of freedom. Degrees of freedom gets slightly more complicated when you're doing a chi-square test with more than one factor, more than one variable. So in this case, it's the number of rows minus 1 times the number of columns minus 1 in your table. So in our case, we've got 2 rows, 2 columns. So the degrees of freedom is 2 minus 1 times 2 minus 1. So it's just 1 actually. So once you know degrees of freedom and you know the chi-squared value, your computer can tell you what the p-value is, and in this particular study, the probability of observing this skew in sex ratio, which correlates with the treatment of the father, probability of observing that under the null hypothesis, which says the treatment doesn't matter, is about 1 in 100. So about 1%, really relatively unlikely. So we reject the null hypothesis, and we conclude that the sex ratio was indeed significantly affected by mate choice. And this is true. So this is what we found in the real world, that female blue tits are able to assess how sexy their mate is and tweak the sex ratio of their offspring as a result. And we find this kind of thing quite often in the natural world. I don't know if anybody's ever looked for it in humans, but it wouldn't surprise me if there was something similar going on in humans. Quite a weird phenomenon, really, if you think about it. Okay. So that's the chi-square test, and it's used for these situations where you've got frequencies that you want to compare to an expectation. It does make assumptions because it is a parametric test, but the assumptions are somewhat simpler than they are for something like a t-test. So really the assumption here is just that you've got a decent sample size, so at least 20 observations in total, and the expectation, the assumption is that your expected counts are more than 5 in each case. So if we just go back to this example, the expected counts here are 47 and 53, so those are clearly greater than 5. If you've got some combinations in your table where the expected counts are very low, then chi-squared tends to perform poorly, and there are some alternative options available, but I'm not going to talk to you about them in this lecture. Okay, that was a long first part of this lecture, so I'm going to stop now, and then we'll come back for some actually a rather easier topic, which is to talk about correlation.